before we get struck down by
the man,
old Mark. Yep, high on his digital hill with his lightning bolts.
Speaking of lightning bolts, did you see that? I don't know if it was volcanic lightning from an eruption or if there was something. Yeah. There was something on top of a mountain that kept getting zapped. Dude, that footage was crazy. I saw it on Instagram.
Maybe someone threw the ring in the volcano or something finally, and
yeah, Zeus was just sneezing.
That did it. I don't know. But we're here. It's Monday. Snakes and Stogies, episode 203. Which is brought to you by BlackBoxCages.com. Check them out. Facebook, Instagram. Use that code THN at checkout. Save yourself some money. Get 10% off your order. If you're in the southeast region, there's a pickup option, so you can save even more. Make the hike up there to North Georgia and pick everything up, and then drive back, set it up, and enjoy it for a lifetime.
I got three 2-footers inbound.
You do. Me and Jake gotta get our stuff together and get our order in. So
I started taking out the tape measure and playing Tetris last night in the snake room, and I don't know how I'm gonna do it.
Yeah, dude, that is always that is what I dread.
Yeah,
it's like at least once a year I gotta rearrange everything, and it's
I think
so flustered when I have to do it because it's.
I've got a good amount of floor space in that room just to kind of like stick stuff in the middle of the room just to kind of move stuff around. And I feel like as much as I want to prep and like, measure twice, cut once kind of thing, I don't think I can. I think the Tetris is just so funky and there's like purple blocks here and red blocks there. And then I got like green blocks in the middle. And I'm just like, you know what, let's just get the cages here and then figure it out. So.
Yeah, I feel like there's the mapping, and then there's the reality of when you actually get them, and you're like, you miscalculated by a couple inches,
Oh yeah, everything's a half inch to the left, and you're like, how did this happen? Nothing fits. And then I think in my mind it was going to fit on a baker's rack. And then I was like, well, wait a minute. If I do that, I can only fit 3 on one rack. I feel like that's a waste of real estate. And then at the same time, I've got all this space on the sides because the baker's rack is 36. These are 24. And I digress. But yes, BlackBoxCages.com. Go shopping, buy them, enjoy them, love them, cherish them.
I just burned my leg.
That'll happen.
It's Monday. So yeah, BlackBox, fullbeastapparel.com still have the buy one get one half off sale going on now until the 11th. So check that out. Everything site wide. Buy one get one half off.
Rock and roll.
And then Cold Blooded Caffeine. Check it out. Best coffee around. Got that Snakes and Stogies blend right there.
Oh, acid green.
The odd combination of Rwanda and Mexico coming together.
It works.
Phil is actually Rwandan, in case anyone was wondering.
Yes, I am.
And I am not Mexican.
Only on my cousin's side. Yeah. Pinky's out.
And then, what is new down yonder?
A whole lot of nothing, man. I never repaired the rinks just because I've been swamped with work and tired, and that's not something you want to do when you're sleepy and groggy. At the same time, last week we talked about how both of us have the ich, the
yeah.
I'm still got it, man. Anna Maria's got it, and
slowly on its way out for me.
Yeah,
that's why I'm glad this kind of worked out because last week I was just
and I thought it might be COVID because when I had COVID many moons ago, it was like that lingering cough for 3-4 weeks afterward. And Anna Maria took a test and she's negative. I did not take a test as of yet, but the more I sit here, I don't think it is. But if it stays, I'll take one.
Last time they had to swab me, man. I thought I started to cry because they like shoved that Q-tip so down, like far into your skull. I started tearing up, and the lady's like, "Are you good?" I'm like, "Yeah." Like,
it's struggling. Always easier when you swab yourself. Insert Jason Kelly joke.
I couldn't even do it. Like, all my tests would be negative because I'd like barely swab it, and then last time I did it, the lady was like impaling me. like it hurt.
Yeah, lost some brain cells. I get it.
A little bit. Yeah.
Yeah.
But it's fine. Like it's fortunately on its way out for me. Now it's just mostly congestion. Everything else I feel fine. So it was not fun.
Yeah. But other than that, man, status quo in Phil's snake world. What about yourself?
Nothing new.
No news is good news.
Yeah, nothing. Still waiting on that pied blood red female to lay. She's cruising like crazy, so it's gonna happen any night now. Maybe tonight because of the rain. We'll see. What are you smoking?
I'm smoking nothing because I can't enjoy it, and it's gonna flare up my sinuses. And
don't be a bitch, Phil.
I know it's a waste, so I'll be enjoying the fine Turkish tobacco of Camel cigarettes.
How you can be able to smoke cigarettes, but you can't smoke a cigar?
Because it's a waste of a cigar, man. It's a waste. It doesn't smell right. It doesn't taste right. Yeah. Maybe if I get a hair up my ass in a few minutes, I'll go up there and grab one.
I don't know. Well, I decided to try something new. I've been holding out on trying it for a while now. It's the is that Foundation Aksum?
Let's see that band. Who's on that band?
I don't know. It's the same guy that's on all their bands. Little
oh yeah, little dude.
Little
Prince.
This is, I think, one of their newer ones that I have not yet had. The first couple puffs are actually pretty damn good. And then because I've been waiting on this episode for a handful of weeks now after our trip to Tallahassee and stuff.
Been chomping at the bit.
Left there with a lot of questions, and the only person that I think could answer them is Miss Jenna Palmisano here.
Welcome to the show.
I mean, how many people are really studying these specifically?
Well, there was a woman in Australia that studied the invasion front there.
Okay.
Yeah, Dr. Crystal Kelleher, but she stopped studying them. The last publication was like 2014. And then Dr. Melissa Miller studied them for her dissertation. And she's mostly focused on Burmese pythons now, but she still obviously is looking for pentastomes and invasive species, including burms. There's a student, a graduate student at UF, Madison Harmon, that was studying lots of different parasites and tegus, and she came across these pentastomes quite often. And then the majority of other people that are studying them are kind of more so studying just snake health in general. So they're looking at a variety of pathogens, and they're not purely focused on pentastomes.
Right.
Mm-hmm.
Well, can we get like an intro for Jenna? I feel like we just
that was sort of just that was the diving in.
That was the segue. Yes.
But yeah, I mean, it can be the long version, it can be the short version. We leave that up to you as far as sort of your background and why you decided to get into these sort of nasty little creatures.
Yeah, they are crustaceans, although we call them lungworms. But yeah, so I'm a PhD student at the University of Central Florida. I'm in Dr. Anna Savage's lab. So most of my lab mates are studying amphibians and amphibian diseases, with one studying sea turtles and sea turtle diseases, which is really cool. I started studying pentastomes actually in my undergrad. So I went to Stetson University and did a lot of research with Dr. Terry Farrell, who primarily focused on pygmy rattlesnakes. He does a lot of stuff with foraging ecology and reproductive behavior. Also does some stuff with like ecophysiology with Dr. Craig Lind. And so I kind of just dove right into research with Dr. Farrell in my undergrad. And we were finding pygmies with pentastomes, these invasive pentastomes. And it was associated with some mortality that we were seeing in the field. And it was quite a bit a ways from the detections by Dr. Melissa Miller. So I was really interested in trying to figure out, how are these parasites moving through the ecosystem? How are they getting into snakes? And so I did a bunch of intermediate host experimental infections. So I took snakes from the wild that had infections and used their feces that had eggs in it to infect other things. So kind of weird, but so I infected cockroaches, multiple species. I infected lizards and frogs and toads, and we also had some trials with rodents and stuff too. But yeah, overall, we did some experimental infections to try to figure out where and how this is moving through the landscape in Florida. And so that's kind of my first introduction into pentastomes and venomous snakes as well. But then I started in my undergrad. During that research, I also worked at a venom lab. And yeah, after my undergrad, I went to the Southwest and
Which
studied
lab?
which venom lab? Med toxins. So also,
Hey,
Hey, that's our boy.
yeah, you know Carl.
Oh yeah,
Oh yeah, everyone knows Carl.
Carl's the man.
Yeah, no, for sure. I mean, yeah, we love Mara. I think.
Yeah, I've been trying to get both of them on a show for 2 years, and they're too
Yeah.
busy. The two of them are too busy.
They are very busy. There's lots of snakes to take care of, for sure. Yeah, definitely. But I've had great experiences there too. I learned so much about snakes from all over the world, their behaviors and stuff. Really enjoyed it. And then I worked in the Southwest with rattlesnakes and Gila monsters. And then I came back here to study pygmies and pentastomes for my dissertation. So kind of a short story there. But I figure people
Yeah.
are more interested in pygmies and pentastomes than they are interested in me, so we can just chat about that.
Well, I was gonna say that's not entirely true. Where did you study, or who'd you work with in Arizona?
I worked with the University of Arizona, and then I also worked for the Bureau of Land Management in New Mexico.
Oh, very cool. Very cool.
yeah. The yeah, so we talked about it briefly like a couple weeks ago, sort of after the Tallahassee trip, but sort of the synopsis to sort of as in terms of like how I ended up falling into this sort of rabbit hole was we went to Tallahassee. We were like the first, I don't even know, it was not long after we got there, I think we decided to go hit just a little plot area that Harry had that he said he had found some cool stuff. So went and did that and looked around. And I happened to look down on this particular spot, and there was a dead pygmy, like a decent sized one. That was just dead. There was nothing, no like physical trauma to it. There wasn't any, like it hadn't been attacked by something. It wasn't outwardly, there was nothing wrong with it except that it was like mouth open, head was swollen. It like rigor mortis hadn't really even set in fully yet, so it wasn't that long that it just died. So I, after looking at it a little more closely, I noticed some worms coming out of the mouth, and I remember the snake talk episode where you had talked about those with Chris and that, and I was like, I wonder if this is those or something else. So I was like, let me at least take some pictures, and I sent them to Jenna, and I said, I don't know if this is what it is or what I think it is, but in case you're wondering, here's the coordinates of where we found it. Like, drop a pin, sent it to her. Took a picture of the landscape. Again, I was like, I don't know what information exactly she's going to want, but I'm going to assume it's going to be just about anything she can get. So I was like, I'll take a picture of the actual field that we were in. The snake itself, up close shots. And that was after we had left that site already. And I think we were back at Harry's. Jenna said, is there any way you can go back and find it again? And sure enough, we went back the next day, and I searched around for probably an hour because, of course, I'm like, I knew exactly where it was in my head, but then I get there and I'm like, wait a second. Was it slightly off to the right or off to the left? Like, I remember it being sort of like a barren patch with some sand, like it was near a burrow or something. But there's like 10 of them out there on that particular plot of acreage. And so after searching around for like an hour, I was on the verge of being like, I don't know where it went. Something must have ate it, whatever, it's gone. And I just happened to, again, look down, and there she was. So took a Ziploc bag, got it, froze it. And then Harry held on to it until Jenna picked her up recently. And yeah, and then after that, after doing the deep dive and stuff and sort of reading about it more, I just had more and more questions, given how much I really do love pygmies. It's a bummer that they seem to be particularly sort of susceptible to them, I guess. But we can get into that. But yeah, I mean, it's weird. So they're called a lungworm. but they're not really a lungworm. Is that?
Yeah, we've Terry and I went back and forth about this for a while because we knew that this was going to be like more and more recognized as a threat to snakes. And so we were just like, all right, we need to not call it Relitela orientalis or Pentastome. Like, it's not very catchy. People aren't going to remember those as common names. So we were like, let's just call it snake lungworm. Everyone's going to remember that. Everyone's going to know what we're talking about. So that's kind of what we went with. But they are crustaceans for sure. They are not worms, though they look like worms. They're crustaceans, and they're actually the oldest parasite recognized by science, which is really.
Oh, really?
Mm-hmm. Yeah, they used to be ectoparasites of smaller crustaceans, ostracods. So they were marine at one point, but yeah, pretty old parasites. They're super diverse. They're all over the world. Some are really generalist, like the one that we're going to be talking about today, and some are really specialized. So there's the smallest pentastome in the world that we know of only infects sugar gliders, and it has a direct life cycle. So they like spit the eggs on like a flower that they're eating the nectar from, and then they'll just consume more of them, or the next sugar glider will consume them. But there's a whole variety of pentastomes, some which can use humans as the final host. But those are most common in the Middle East.
Okay. And so there are native ones, but are those more the actual worm variety, or are they the like crustacean-ish ones? Is that?
So are you asking if I guess so. I'm sure there are a bunch of things that we call lungworms, including like there's one that's specifically like a nematode that we call a lungworm. But when I'm talking about lungworms in Florida, snake lungworms, I'm talking about pentastomes overall. And so there's two native pentastome species that commonly infect snakes. So yeah, there's two native ones, and they are the crustacean pentastome.
Okay.
Yeah.
Okay. Mm-hmm. So the issue isn't necessarily the native ones, because obviously over time those species, our native stuff has adapted to be able to manage those and sort of handle them and just live with them to some degree or another. But it's the invasive ones that we're talking about tonight that are a different story.
Yeah. So pretty much, exactly. I mean, one of the native ones is Chirocephalus coarctatus, and they're mostly going to be using fish as intermediate hosts, and so they're often infecting Nerodia. That's who I see them the most in, so water snakes. But usually, like, water snakes will only have a couple of them. But the other one, Porocephalus crotali, is one of like the most well-studied pentastomes. Like, we know a lot about its life cycle. We've done not me, but like in the 60s and 70s, they did experimental infections with cottonmouths and rodents and stuff. So we really have a good handle on Porocephalus crotali. But some of those infections can get really intense. Like in cottonmouths, I've definitely pulled out, like 12 really chunky parasites. But yeah, it hasn't really been associated with any mortality events of wild animals. But then again, I don't really know how much people are looking at the impacts of native pentastomes. I think the invasive pentastome is probably more of a threat given all of the other threats that our snakes are facing in Florida.
Do they know how long the invasive one has sort of been floating around? Because it seems like just within the last few years, it's kind of become more of a concern or a concert, like a conversation being had. It's not like they just kind of showed up out of nowhere and all of a sudden they're just across the state, overnight. So
yeah.
You mentioned 2014, they were doing some studies on them and stuff, but I mean, do they know how far back they've been documented or recorded? And
yeah, so I mean, I think the issue with like a parasite like this too is like we're likely not going to detect it until it's saturated in an ecosystem, and then it's kind of like, okay, we're obviously going to find snakes with worms crawling out of their mouth if the prevalence right now is 30 to 40%. When it's just starting to spill over from the pythons, like I don't think people were going to be able to find that easily. They didn't even know it was a concern. So I'm guessing that it's earlier than what we know, but I do know the first snake that Dr. Melissa Miller found was, I believe, a garter snake in 2012 in South Florida. But given how long berms have been here, I'm guessing that that spillover event probably happened earlier. We just aren't recognizing it until it's like fully saturated ecosystems. And, you know, we're starting to see a lot of snake communities crash overall, and whether that's related to habitat loss, degradation, invasive species, other pathogens, this is just kind of the nail in the coffin in a way, unfortunately. So I think, we're just starting to recognize things as it kind of saturates the ecosystems moving up the peninsula.
Is there a what's the word I'm looking for? A current distribution map, or a believed current distribution map of where the crustaceans are most infectious?
Most infectious, no, but I do have a map of like all of the counties that we've found them in.
Okay. Is it mostly West Coast Florida? Is it more centralized? Is it just the whole state?
Am I able to show you?
Yeah, actually, if you send it to Smitty or we can throw it up on the screen.
Either that or the present button down at the bottom should give you a share screen option.
Yeah, there you go.
Oh, yes. Okay, cool. Okay.
I think we should rename it Long Crab.
Can you see that or no?
No.
What the heck? Lung crab? Yeah, no, that one, I do kind of like that. Oh.
But I can also share it if you need me to, if there's something in
Google
particular.
does not like me to do that, but basically it's everywhere in the peninsula, and we've just found a new record in Madison County. So it's starting to move northwest now. But the farthest north that we had found it before Madison County, which we found a couple weeks ago, was Duval County, which is just one county away from Georgia.
Oh, wow.
Yeah. And also Madison County borders Georgia, but everywhere else in the peninsula, east, west, central, we're just kind of starting to fill up the map now with more people looking.
And has there been a case of a specimen found who has the lungworm and is surviving and living, or is it a guaranteed fatality, just a matter of time?
We don't really know. Yeah.
Okay.
There's, I guess, like the only way to for sure know if it can cause mortality is to do an experimental infection on a healthy snake to be like, okay, we know that it can cause death. My guess is that it probably depends on the species. And just how many it has. I think, Pentastomes have evolved quite tightly with a lot of their host species so that they cannot be detected by the immune system. So they cover their cuticle in surfactants that match their host lung surfactants. So they do a lot of things to try to reduce detection of the host immune system. So yeah, but I don't know if that works the same in a novel host. But we don't know if it's definitely fatal. I mean, I think it depends on the size of the snake, the amount of worms in the lung, and then also just kind of like the state of that snake before it getting infected. Okay. And it could also have to do with kind of like the metabolism of those snakes, where like you see a black racer, like what actually caught a black racer outside of my house 2 years ago now that died and had pentastomes. Okay, that map's old, but yeah, so we've got a lot more in there. I think there's 27 counties now.
Wow.
Oh, wow.
Yeah, so since that map, we've added 13 counties.
Wow.
Yeah,
I'm assuming they're all ones that are kind of headed this direction.
So we actually, so Madison is kind of just northeast of like the armpit. So going the other way, like around there. But most of the stuff that we've got is in Peninsula, Florida. Hopefully it moves into the Panhandle slowly or not at all completely. But
what are the color codes? Just because it's kind of small on the main screen.
Yeah, no, those are just different publications.
Oh, okay.
Yeah, people that found each one.
Oh,
yeah,
okay.
I can. If you go to the SLAM website, there should be a better map. I can send you the link real quick in the chat. But yeah, so what I was saying is like you see black racers that are doing well. They've got plenty of fat bodies. They're yolking up follicles, and they have like 60 plus of these pentastomes in their lung, and they're seemingly fine. Versus I've found 4 pygmies in the past few months just dead in habitat with infections. Also, they also had other pathogens too, like snake fungal disease. But yeah, we just don't see that type of mortality in black racers. And maybe they can deal with it because they're eating so much all the time. And something like an ambush predator might not be able to handle that.
Yeah, that makes sense. The
yeah, and I mean we also have to remember that a parasite that doesn't survive in its host for an extended period of time doesn't live very long. Like that doesn't work. It's not how they operate. So I would imagine with those in particular, like it is kind of a numbers game where it's like, I guess when the bus is full and there's no more space, that's when you get the most problems, I would imagine. But
yeah,
again, I'm just a normie. That's just my brain.
Has there been a well, go ahead. Sorry.
I was just going to say, like, it's tricky because like some of the pentastomes, we don't really understand their biology or their evolution to the point of understanding competition. So like if those pentastomes aren't related in the lungs, like there might be different interactions than you would expect.
There seems like a lot of moving parts, a lot of sort of things have to be happening to.
Yeah, there definitely is. Yeah, the Madison County is just not on that one yet, but everything else is there.
So what is the SLAM findings?
So SLAM is Snake Lungworm Alliance and Monitoring. So that's the group that I created with Dr. Terry Farrell 2 years ago now. And we've got about 94-ish collaborators across the southeastern United States that are either picking up road-killed specimens that they come along, or if they're permitted for research, handling snakes, they collect cloacal swabs or fecal samples so that we can check for eggs in live snakes. So that's how we've kind of been able to track more of the spread, both like taxonomically and geographically, is just with all of those collaborators.
Excellent. Have there been a specimen in captivity that has, how do I phrase this, survived long enough to pass the parasite? Or is it stuck with it for life? It's just going to keep reproducing inside it?
So I don't think we truly understand reinfection. There was like a paper on a cobra that they thought was self-reinfecting, but that was like, it was one of those dissertations that was so outdated that I was like reading a printed file on my computer and the font all doubled and stuff. And I was like, I can't do this. But so we don't really know much about reinfection. We do know of pentastomiasis in the pet trade. Lots of different species are in the pet trade, including the invasive one that we're talking about today. But they live a pretty long time, like on the order of years. We know the native one that we know so much about can live over 6 years in a snake's lung, which is kind of crazy. It's likely that the invasive species lives a shorter amount of time. And my guess is that the snakes are probably not reinfecting themselves, just based on the life cycle of the parasite. But we know, like there was a banded water snake in Michigan that was found with an infection. Terry actually found it and published on it. And it was kind of, snakes don't cough because they like don't have a diaphragm, but like it was kind of like puking up pentastomes.
Wow.
And that's how he found out, because this lady posted on Facebook and was like, does anybody know what this is? And he was like, I do. But the three cases that I've found in the pet trade, all of the snakes that were sold were wild caught from South Florida, and they died within 2 months. They all became anorexic. They wouldn't feed. They got skinny. They started to spit up like white mucus with eggs in it. And then they died, and they had infections from 3 to 39.
Wow.
So yeah, I haven't had a lot of cases where I've read about where snakes are just living with it. I know there was this one paper about, I feel like it was a python. I don't remember where in captivity, but they were giving it, what is that? Ivermectin. They were giving it an anti-helminth, and they thought that it was working. But I don't think, from the vets that I talked to, anti-helminths won't work because they're not helminths. They're crustaceans. Yeah.
What were the species? You said three different species
in captivity
that were field collected in South Florida that wound up dying.
Yeah, so there's 3 individuals. 2 of them were garter snakes, and then one of them was a scarlet king snake. And then I most recently got an animal that was taken from Southeast Florida. It was a Florida king snake, and that one also stopped eating and then died. And it had 18, I think, of the invasive Pentastome in it.
What's considered like a really heavy load? Like, what's considered a lot?
I would probably define
in general. I know between species it's going to vary because size and stuff like that. But
yeah, so I mean, I would say like a lot is also dependent on the species and the size of the snake, but the most that I've seen is 107.
Wow.
Yeah, 107 in a black racer. Pretty crazy, from the west coast of Florida. Yeah.
Yeah, that's wild.
He was not in good shape. He was definitely super emaciated. Yeah. Some of the snakes that we see, like I was talking about black racers, maybe not showing that many external symptoms, but there's been a lot of cases where black racers are kind of found acting weird. And then they're found dead the next day, and they have pentastomes. Yeah. So there's some behavior stuff that people have observed before snakes die, which is interesting.
Yeah, and that was something I had asked you a couple weeks ago because you had posted about a garter, and it made me wonder if there are certain species that seem to be where they're more prevalent in them because they are more lizard, frog, snake eaters and things like that. Is anything getting these from like rodents? I mean, do the invasive ones, are they in rodents? Like, do they, even if it's like a temporary thing, what's the?
So that one's a hard question to answer because there's just not much evidence of Raleigh-taila orientalis, the invasive snake lungworm, in mammals overall. The only evidence we have of that is a Kenyan shrew, who was an intermediate host that had larvae in it. Other than that, we don't really have any evidence in mammals, but it's likely that they're utilizing mammals as intermediate hosts. But from the Australian invasion, as well as the North American invasion, the highest prevalence is among snakes that are eating frogs and lizards.
Wow.
But I mean, it's likely that they're using mammals. We just don't know much about their role in the life cycle.
Yeah, the primary reason for me asking if it's quote unquote passable is because before the show we were saying I go field herping quite a bit, and I have not yet seen a dead snake with like Smitty had with worms coming out of it, and I usually check most roadkill and stuff. And
I will say real quick, that was very subtle. Like there was one sizable worm that I noticed, and it wasn't until after sort of really looking at it for a few minutes that I noticed it. And I was like, well, let me snap a picture of it. But we'll get into that in a second too, because we talked about it briefly before we started, before my internet bit the dust, that it was not what it seemed. But continue.
Yeah, and I look forward to that. Excuse me. I have 2 pygmies from my county, Palm Beach County, and both collected in very similar, say 5 or 6 mile from each other. I've had them about the same time. One of them just crashed like horribly out of the blue, like lost its weight, skin showing just real quick. And I was like, how does this happen? What is going on here? And like, I didn't see any kind of symptoms or nothing. And then it slowly just came back to being good. And this is long before I knew about these pesky crustaceans.
Yeah.
And seeing your work and Smitty showing me your stuff and Instagram and him telling me the stories, I was like, man, I wonder if that's what that was. But I almost feel like I would have seen something or noticed something, or probably the snake would have died if it had got that far.
Yeah, I mean, when an animal is stressed, those pentastomes do tend to move, and so that might have stressed them even further. But yeah, or it could have, I don't know. Yeah, I would definitely I'm happy to look at poop samples.
All right, I will try to procure you some.
Yeah, I mean, I'm sure. How long have you had them in captivity?
God, probably 4 or 5 years, and
Yeah, it's been a while.
this particular one was about a year ago.
Okay. Yeah, I mean, and you're not feeding them like are you feeding them wild caught things?
No. I'm not a big deworming guy in terms of field collected animals. I figure if they're that healthy in the wild, I just need to keep the stress down, keep them healthy, and things will go their course. I mean, I have dewormed stuff in the past with vets and without, but the pygmies, I don't. I just leave them be, and they've been great. And then that one, just something snapped, and it was almost over the course of like 2-3 weeks, and I was like, what's going on with this? And then I wound up keeping more tabs on it and floating in some water to stay hydrated. I thought maybe it wasn't drinking, maybe it was something with the water bowl that made it not want to drink or whatnot, and misting, and maybe it was too humid, maybe it's too dry. You go
heat
through
rock.
Yeah, it was the heat rock.
Damn heat rock.
It was the easy bake oven. And then it just, I'm 99% sure that one is. Okay. Yeah. Why do you ask?
Well, I'm just wondering like what time of year that happened and stuff, and if it aligned at all with their reproductive cycles. Because
I would say it's probably too young at the time. I got the thing. I caught it when it was about I would say maybe 2 or 3 years old when I got it.
The females will start reproducing around then.
They're really small though.
Oh, okay. They're body size wise,
body size. Because I mean,
how thick?
I would say your pinky. Well, I don't know how big your hands are, but
yeah, I mean that's a small snake.
yeah. Because I mean, don't get me wrong. I've produced them in the past, and we're talking cocktail straws.
Yeah.
As babies, and feeding them pinky parts.
Yeah.
So this one was not that, because I don't field collect that, but yeah, it was I would chalk it up to being 2 or 3 years old just from all the pygmies I've played with over the years. But who knows?
Yeah, I mean, my guess is that if she was that small as a female, she was either not doing well or because I feel like she should be bigger by 2 or 3 years old. That sounds more like a 1-year-old snake, but I'm used to some monster pygmy sites, so it's hard to compare.
Yeah, you're definitely seeing a lot of them. Good Lord.
Yeah,
I think I've seen around 360 since last June.
Wow, that's a lot.
I put in. What was that?
So I've seen 2 dead ones.
Oh no! You can come out with me. There's definitely sites where you can find live ones.
Everyone keeps telling me, I got a site. We're going to find them. And then it just never happens. So
yeah, well, I have to find them. My research depends on it. I have to put in a lot of effort for sure, especially at some sites. But been looking for pygmies for a while.
Are you? How do I phrase this? Are you pit tagging any of them? Are you doing any kind of photo record of patterning or anything to kind of ensure that you're not looking at the same one over and over again?
Yeah, so the major part of my research is actually looking at the genomics of pygmy rattlesnakes across Florida. So I pit tag all of them because I'm taking genetic samples. So I definitely need to know who they are. Yeah. If they are too small, I'm not typically taking a genetic sample, but if I do, like, say I'm in a national forest and I'm low on samples and I know it's like I need to get every pygmy I have here for genetic stuff, I'll make sure I get a really good picture of their head in the squeeze box, just so I know later on that I didn't get the same snake for the genetic workup.
Yeah.
Mhm.
Yeah, but yeah, I'm pig tagging them. I use like typical like biomark 8mm tags with a 16 gauge needle. Yeah, I do go into the body cavity with pygmies because a lot of them are just too small, and there's evidence that they can break them if it's subdermal or subcutaneous. So I don't do sub-Q pit tags. I go into the body cavity.
Wow, that's awesome.
What's the population densities like in the spots that you're? I mean, it seems like so I got there's that little book, the quote unquote glorified pamphlet, me and Phil call it, on pygmies.
The diminutive giant.
Yeah, and it talks about just how dense some of those populations can be, but it's mostly just due to their size, not necessarily like in a obviously in a certain amount of space with a small animal that doesn't really have a very big range. Like you're going to have sort of concentrations of them in certain areas. What are the?
Yeah, I think it more so has to do with like pygmies are somewhat generalist. Like you can find them in sandhill habitat. You can find them in uplands. They're just not going to be as dense as they are in the swamps. I typically get a lot more pygmies when I'm kind of in a transition zone from like a very low wetland into a more mesic hammock. That's where I find the most density of pygmies. And then there's just certain sites that they seem to like really have hotspots in. But yeah, usually I have the most luck kind of in those transition zones from very swampy to more mesic hammock.
Very cool.
Yeah, but you kind of can find them anywhere. Like you can be cruising sandhills and get one.
Well, it's
but-
harder and harder in Southeast Florida. I'll tell you that.
And I mean that's
Yeah.
from what I've seen and talked to, as far as Harry and talking to Tino a little bit when we were down or up in Francis Marion. It seems like the sort of the sandhill areas seem to be where a lot of people are seeing them, because I'm in a really swampy area.
Yeah.
Granted, I'm on the barrier islands, and I'm still convinced that the island I'm on in particular, pygmies are not on it, because I've never seen a DOA, never seen a live one, like nothing, or DOR rather. But even sort of off the island and in the actual mainland, I guess, of South Carolina, like I still haven't seen any. Like we've had to go to inland to the like Okatee Hunt Club area, which is not crazy far from here. It's it's 30-45 minute drive maybe. But just here, like directly on the coast, it seems like I haven't seen any signs of them. But
Yeah.
maybe I'm
I don't.
trying
I feel like
hard because I can't see
Yeah.
corals either, and they're everywhere. So
I found a coral the other day, not to
I
make you
saw.
upset. Yeah, no, those are usually just like such random bycatch. Like when you go out so much.
How often are you going out?
Well, now that the semester is over, I go out like 5 days a week.
I was to say, it looks like it's about daily.
Yeah, like 4 to 5 days a week. And then during the semesters, like when I teach and stuff, it's like, well, now I don't. I just finished my classes, so you have to do like 2 years of classes as a PhD student. So I just finished those, so I'll probably continue to go out, 3 to 4 days a week when I'm teaching. I have to do a lot of lab work too, so I have to make sure that I'm here doing that and not getting behind on lab work. But yeah, I go out quite a bit. The goal is to get 1,000 dusky pigs by the end of the study, and kind of just have a good spread of sample size across the board, just to kind of have an accurate representation of what they're doing, how they're doing, and what the genetics look like. But so I have 13 locations that I survey, 3 of which I hit 2 days each month. And those are kind of like my heavy mark recapture locations that all 3 of them have Pentastomes and snake fungal disease. But each of the pathogens were detected at different times at those locations. So kind of really interested to look at how co-infection with snake fungal disease and the invasive pentastome impacts survival and growth rate, and kind of which demographics of the populations are hit the heaviest from those 3 sites. And then the additional 10 sites kind of spread across the entirety of Florida, like all the way west in the Panhandle, all the way down south to the Everglades. And those are to get an idea of what the genetics look like and like how many distinct populations we have for better management in the future. And I'm really interested to see if there are adaptive immune alleles related to the severity or susceptibility to diseases. So I'm looking at adaptive immune alleles and kind of comparing that to neutral genetics and disease severity and all that.
Is there any major correlation between the ones that have both, as far as snake fungal and pentastomes, and or is it more of a population thing with the SFD?
Yeah, we don't know yet. That's like one of the things that I'm really interested in figuring out is like what's going on with these co-infections. I mean, pygmies in Florida, some of my sites have really bad snake fungal disease, like, and they don't have pentastomes, or like some of them don't. Like one of my northern sites, I've detected pentastomes in other species there, but not pygmies. And like some of the SFD lesions there are really, really bad.
Wow.
But SFD is more seasonal, but the prevalence is really high, and there's definitely a lot of like sublethal impacts on the snakes. Like it changes how much reproductive hormones and when it kind of alters their reproductive cycles.
Really?
Yeah, it alters their basking behaviors, which I'm sure you knew. It alters their metabolic rates, and so they're burning more energy if they're sick. It impacts their evaporative water loss. So it's like, okay, they're dealing with some severe seasonal issues with snake fungal disease, and then on top of that, they could have this.
Getting it from all fronts.
Yeah, that's really, I think, my biggest concern is kind of the combination of all threats. Yeah, I'm sure if there was plenty of habitat, there wasn't hogs or other invasive species or other emerging pathogens, that pentastomes could probably, the invasives would just be here and not have much of an impact.
Yeah. The majority of your sites, is this state wildlife management areas? Is it state park? Is it federal property? Is it agricultural?
Everything.
Okay.
Yeah, so they kind of spread across national forests, national parks, state forests, WMAs. Yeah, the whole bit.
Okay. Yeah, the majority of my pygmy spots I have by me is in between agricultural land.
That's what I figured. Yeah.
Yeah, and the WMAs that are nearest to the city, there's no pygmies. It's either way too wet, or there's just so much human activity, and it just finding them is damn near impossible. But the agricultural areas still have a lot of good pigs. But I wonder if agricultural runoff and pesticides and herbicides are playing a factor in the fact that they're killing some of the bugs, perhaps? Or is it just wiping out the whole population of everything?
Yeah, I don't know. I mean, I've heard of some agricultural pygmies being quite big, so perhaps they're eating mammals. But yeah, I'm not really sure. I don't know. Yeah.
Here's a question in terms of sort of ontogenetics and stuff as babies. Like, what are they eating? I mean, they're tiny. Like Phil mentioned, as when they come out of mom, like they are teeny tiny. Like
they are literally like the smallest pygmy I've ever found was in the Panhandle, and it was 2.5 grams. It was so small. It was teeny. But most of the time, the neonates that I find are like the weight of a quarter, so like 5.6 grams. From what we know and what we understand of pentastome biology, it's not likely that the mom would be able to give them pentastomiasis. And we don't often, I don't see infections in young pygmy rattlesnakes. I just don't think they're consuming big enough prey items or enough.
Well,
prey.
that's why I'm wondering is it a ontogenetic thing, sort of like we see in some other species like green tree pythons, where they start out eating one thing and then as they age, it shifts. Like there's a pretty big shift in preferred habitat, but also what they're eating. Because I'm assuming with pigs being that small when they're babies, I mean, they have to be eating, I would think, mostly insects and then maybe like fresh Anolis babies, like
small frogs, man.
Yeah,
too.
small frogs, baby skinks. You see a lot of like baby skinks start to come out when baby pygmies are out and about.
Mhm.
So skink would probably be a good bet too, but they definitely will eat inverts as well. But yeah, and like the teeny frogs, like if they're eating like a grass frog or something, like the grass frog's not likely to have pentastomes because the life cycle starts with a larger invertebrate like a cockroach that's going to be feeding on the feces of a snake. So yeah, it's not likely that too many younger snakes are going to be infected.
Wow. Yeah, because the one that we found that first day, I mean, that was a large, I wouldn't say she was like massive, but she was a decent sized adult.
Yeah, she was normal for sure.
You mentioned that there were ovum.
Yeah,
when you looked into her.
Yeah, she had 7 or 8 developing follicles. Yeah, you caught her in March, I think it was.
Yeah, it was the end of March.
Yeah, so that is typically when females are vitellogenic, so they're yolking up follicles, and then they'll fertilize in April with the sperm that they stored from the fall.
Okay.
So those were probably follicles, and by now they would have been ova.
Mm-hmm.
Yeah, and then they're like full-blown pregnancy in June, which is kind of cool to see. They look very plump. And they're usually easier to find in June because the females in early pregnancy are in ambush quite a bit. They're eating a lot.
And is there any relation there in terms of females having a heavier load with the pentastomes as compared to males? Because I would think if they're eating a lot more like that, then the likelihood of them taking on more of them is greater.
Yeah, we don't know yet. I mean, we do have quite a bit of sampling across like DORs, there's kind of like 4 main leaders of SLAM, and each of us have dissected like 200 snakes in the past couple of years. So we could probably look more into that, but I think it's kind of evenly split so far, at least what I'm seeing in pygmies. But we haven't really like analyzed or assessed the data in that way, just because we don't have enough of the sample size from each species. But I could definitely start to answer that with pygmies for sure.
Well, I mean, again, it's another variable where it's gonna, like, even by location, I'm sure it varies because the food availability is going to be higher in some areas than other places. So it's just, again, so many moving parts. It's like a really fucked up Rubik's cube.
Yeah, no, there are a lot of moving parts for sure.
Our good friend Lisa in California wanted to know, she's sorry that she's late. Maybe we've already gone over this, but are pygmies getting it the hardest?
Oh, that's a tricky question to ask.
Because obviously it's difficult for you because you're focusing on them and you're actively pursuing them.
Yeah.
But in your travels with bycatch and whatnot, is there an equal ballpark ratio of other critters to pygmies or no?
So are you more so asking if I'm seeing declines in pygmies aligned with the arrival, or
basically are pygmies more susceptible? Are they dying more from the pentastomes than other species are?
There is empirical and anecdotal evidence that points towards yes, but I would be uncertain to jump on that and say yes immediately, especially because that's one of the things I'm really interested in, is looking at the genetic underpinnings of susceptibility. So I just don't know. I think we need to look from like molecular levels up to understand susceptibility. It's kind of a tricky question, but I mean, from the evidence that we have, there is data showing pygmies declined that like really dramatically that align with like temporally with the arrival of Relicella orientalis, the invasive snake lungworm.
Wow.
And we haven't really seen direct evidence for that in many other species, but we have seen communities just crash, and it is kind of aligned with when we first detect the pentastomes there. But there's lots of moving parts, especially here in Central Florida, where snakes are losing their habitat rapidly. So it's kind of hard to just point fingers at this parasite. Yeah, it was a long-winded answer of.
That's perfect.
I don't know.
Yeah, it's a very good. I don't know.
Yeah.
The female that you picked up from Harry, you said that was not the invasive.
Yeah, so this is actually really interesting, and I don't know what to do with this information just yet. But as soon as I picked up that female from Harry, I opened its mouth and just kind of pinched and looked in, and I was like, that's Porocephalus crotali. The body shape is a lot different. The cuticle is a lot different. The head shape and the hook orientation is really different. The invasive one kind of has more of a triangular head with the back hooks being larger and typically kind of like flared out. And then the Porocephalus crotali has like this like kind of just like club-like head with the hooks like all in a line, and they're all the same size. And it had about 18 of them. And the reason why I don't know what to do with this information is that we've never found a native pentastome in a pygmy rattlesnake before. So this is like a new host record, but no one's really looked at this stuff. So it's kind of hard to say that this is like, obviously it's not like, whoa, this is the first it's ever happening. It's like, no, this is just the first that we've recognized it. So like, cool. The invasive pentastome is not in Leon County. But yeah, no, pygmies can get native pentastomes, which might be beneficial in the long run for tolerance of Relicella orientalis. I mean, unfortunately, from what I'm seeing in the field, that might not necessarily be the case, but yeah, so that was kind of bizarre, and I can send you the pictures of the dissection. She had, I believe it was 18.
And look, Smitty, you're going to get in publication.
Look at that.
It's fascinating.
Like I said, I'm just a normie with a Ziploc bag, and I found a dead snake.
That's how we're finding pentastomes in New County.
Hey.
Yeah.
She's like, can you go get the body? I was like, I'm going to try for science.
I felt bad. I was like, please, please go get it.
Well, the frustrating part was like Jake and Harry were like, they wanted to go to like a couple other spots before we got there. And I'm like, we really need to go there like first thing, because something's going to find it and eat it if it hasn't already.
If it wasn't fire ants.
We got super lucky. Yeah, and that was sort of my biggest concern, was like ants are going to be all over that thing. Mm-hmm. There were none. Like she was still just sitting there like when I had found her, you know.
Wow.
Strange.
Yeah, it's super strange. Yeah, it's incredible information, so I really appreciate it. It's just fascinating because we haven't seen that, and we've necropsied quite a few pygmies in the past handful of years. So
do the native ones operate the same way in terms of like how they attach themselves and overall, like
no,
how they get in there and do their job? Is that
how they get in there? Yes. Like, well, from what we know is basically like a pygmy eats an infected prey item or a snake. And then the larvae just bore straight through the viscera. Like they just make a direct path and like cause a bunch of inflammation and stuff, but they literally just go straight to the lungs, and they'll hang out there and they'll molt several times with Relicella orientalis, or with pentastomes that we know the life cycle of. I think it takes about like 80 days for them to reach the sexually mature molt, and that's when they start to reproduce. So basically they have internal fertilization. So they'll copulate, and then the female will store the sperm in the spermatheca, and she'll go through like one final molt, make sure the uterus is all developed and stuff. And then the males just kind of hang out for a bit, and then they die right before like 200 days or something. and the females just keep pumping out eggs. But the reason why I said no, there is a difference between the native and the invasive is the two natives have like really club-like heads, especially Chirocephalus. Chirocephalus literally looks like a lollipop. It's the most bizarre alien creature I've ever seen.
Wow.
And they're huge. Yeah, I can send you some pictures, or actually I put together a dissection video to show the difference between them. I'll send you my YouTube.
Well,
But yes,
while you're
yeah.
doing that real quick, you'd mentioned, so they're not self-fertilizing. There is male and female.
Yeah.
And they're copulating
Self-fertilizing
in the lung.
Yeah, they copulate in the lung. Yeah. So
And then where do those
this
molts
is a house
go?
of
The
learning
molts-
doctors.
The molts are just in. What's so funny, man? I thought it was a viable question.
Asexual, I think is the term you're looking for.
Well, no, because you can have asexual animals that can't fertilize themselves, like land snails, right?
I mean, I guess I don't.
Anyway, what I'm saying is, so you've got boys and girls breeding inside the lung, and at the same time, where do those molts go? They're just in the lung, just there.
Yeah,
That's
so
horrible.
it can cause so pentastomiasis can cause pneumonia, can also cause sepsis because they are creating a lot of waste, and then they're just dumping the eggs in the lung as well.
Oh wow.
But yeah, the native ones really kind of bore into the lung tissue, and so they'll kind of like encapsulate themselves with hardened tissue, versus like the native one just kind of like slaps on the side a little bit. So if there's a few videos of like us pulling them out in dissection, you can see that, but there's the only way to kind of treat pentastomiasis is endoscopy, so physically removing the pentastomes from the lung. And you can see it, like Dr. James Wellahan has a couple videos, like you have to like tug on the native ones to get them out, and then the invasive ones just kind of like come right off. Yeah, it's
super cool.
Yeah,
scary but cool.
Yeah, I won't tell you about the human stories that I've read about.
Oh, now you gotta. Now you gotta.
You're gonna get grossed out.
No, hardly. That's what the show's all about.
Okay. Well, like I said, there's one pentastome species that we know of that can infect humans as the final host, which means we get the adults. This one primarily infects mammals in the Middle East and in Africa. And it's called Linguatula serrata. And it mostly, again, uses mammals. So they find it in lions and stuff, but it's often in canids, and people can get larval infections just by hanging around dogs or other things that have it, because the nasal secretions will actually have the eggs out, and so they lick your face, and then you can get a larval infection. Larval infections typically don't really cause symptoms, but if a human were to eat raw sheep or goats or something else that was infected, they can get an adult infection. And so Linguatula serrata actually lives, instead of in the lungs, it lives in the nasopharynx cavity. And so there's a story about this guy that had a really bad headache for a very long time, and he like kept seeing doctors and stuff. It is a publication. It's not just like a story.
Right.
And he just sneezed really hard one day, and they came out of his nose, and he was like, oh, so that's what had been giving me the headache, which is kind of like absurd. But yeah, it is most prevalent like in the Middle East, where people are eating raw sheep and stuff like that. But yeah,
that's nowhere near as graphic as I was expecting.
Well, there's also stories in like Southeast Asia and in Africa where rural farmers will be eating snakes, and like that is primarily how people get infected with a different genus of pentastomes called Armilifer. So Armilifer and Linguatula are kind of like the main genera that impact human health. But a lot of times people, like there was one case that was kind of gross, but people eat raw snakes sometimes, and they were talking to indigenous peoples, and they were saying like they just thought that the pentastomes were like part of the snake, so they would just like chew on it, feel it, and then just spit it out. So people can get really intense larval infections, and it can, if it like gets really bad in certain organs like the liver or something, it can mimic liver failure and stuff like that. But that's like typically, from what I've read, it's very rare for things to kind of go awry like that. But yeah, there's definitely been infections found in humans all over the world, which is very interesting.
Yeah, and I know that anyone who's listening to this, they're gonna wish I asked if I didn't ask. But what are the odds of us crazy people that keep exotic animals at home, contracting it? I imagine it's fairly slim.
Yeah, it is. Yeah, I mean, if you're washing your hands, you're fine. If you're not putting your snake's poop in your compost, you're fine.
Yeah.
Yeah, you would have to ingest snake poop somehow,
Right.
to become infected. Yeah, which I mean, like, if you're a casual handler and you're just like, I don't know, snake pukes on you or a snake poops on you and you just kind of like forget and go eat a sandwich, I guess it could be possible.
Is there a way to make this full screen, Smitty?
I'm trying here.
There we go.
I think
I'll also give a disclaimer that this is for educational purposes. This is medicinal.
YouTube
and
doesn't care, dude. They're gonna demonetize it regardless. But who cares?
I just don't want them to cut the feed like they did last time.
They're not gonna. I don't think they're gonna cut the feed. Did they cut the feed?
They cut the feed when we did that other necropsy thing.
That was a long time ago. I think they've gotten better now.
Yeah. All right. So, do you mind giving us a little play-by-play?
Yeah. So this is a Nerodia. I believe it's a banded water snake. In these, so he's in the anterior portion of the lung. This is Terry doing this dissection. A lot of the times after the snake dies, the pentastomes can kind of burst out of the lungs, be in the trachea, be in the mouth, be in the lower organs. This here is the invasive species. This is Reptile orientalis. You'll see they just kind of like pull straight out. They're not stuck in the lungs or anything like that, but they fill the lung cavity, as you can see here. And all these big ones are females. The males, that might be a male up front, but the males are usually smaller and the females are quite big to fit all those babies in there. So yeah, Terry is just pulling out some of those. This one's a pygmy.
Mm-hmm.
It looks like there's a pretty small guy in there. Obviously, they don't get that big in pygmies. But yeah, so again, anterior portion of the lung. I think that's a male that was like on the paper. Yeah. Okay, so this one.
Now, at this point,
yeah.
Are the worms dead at this point, or are they still active and alive?
Yeah, these were frozen thawed roadkill snakes. So, but yeah, I've opened snakes. Like one of the ones that I found, a pygmy rattlesnake at one of my sites, got run over by someone on a bicycle. I don't know how they hit it. Whatever. Anyways, it had the worms. Like I've picked up snakes that had live worms in them. I think this one shows Chirocephalus coarctatus. So this is the one that has the chunky lollipop head, which you'll see. It's like going to be thickly embedded or deeply embedded.
It's really
you'll wait till you see him like pull it out. Yeah, so these guys I've only seen in Nerodia, and like they just have lollipop heads.
Yeah.
Yeah, see, it's really in there. But yeah, so these guys are mostly using fish as intermediate hosts, and they've got that big lollipop head. And then I think the next one might be a cottonmouth with porocephalus, which are kind of just like blobs. I might be talking about something. I forget. I made this video a long time ago. Yeah, they have weird. Yep, that's porocephalus crowleye next. Yeah, so that's a cottonmouth. I don't know why I'm telling you guys that you knew. I'm like, that's a cottonmouth in case you didn't know. Yeah, porocephalus crowleye, these ones are gross. All of the coils in there are actually like, that's her uterus. And so,
wow.
Yeah, they have like a highly like extensively coiled uterus that just fills their entire body. And they they can produce way more eggs.
Maximum damage.
Yeah.
Yeah. They can produce way more eggs than Reptile orientalis can. That's what they look like when they're kind of fixed in ethanol. They turn quite white. But the ones that came out of the pygmy that you found with Harry, those look just like this. Yeah.
So like when you find them, like if you find a dead snake like that and you see worms coming out of the mouth, is that the worm literally trying to get out of the body, or is it?
So
like, are they trying to leave to hopefully find something else?
That's what larvae look like. They kind of look like tardigrades. Yeah. Yeah, they have those hooks like throughout all of their life stages. Even as eggs, you can see those hooks in there, just because they got to be ready to go and attached to the next host. What was your question? Oh, yeah,
leaving the body.
Yeah, we know that like when snakes get stressed or when they're dying, we know that the pentastomes move. We don't really know what their goal is for that, but they are relying on the host for oxygen. Like they don't have full respiratory systems. So it's likely that they need oxygen and they're just trying to get out. But there's been a case where like my friend was handling a black racer and it got stressed out from the handling and it just like puked up a pentastome on his arm. So yeah, I don't really know. My guess is oxygen, but I'm not really sure.
Yeah, because you're not entirely sure about sort of the pathology is the word for it, like what's actually killing the snakes, be it anemia or just lack of appetite, not eating and starving slowly, or maybe a combination of all of it, I'm sure. That's
kind of like what we call like the disease mechanism. Like how is this actually causing disease in the animal if it is?
Yeah. Do they know how long an adult would survive outside of a host?
I don't think we know, but I've seen like the black racer that I found. She was dead for quite a bit, and the pentastomes were like moving on the cement. And then the snake that got hit by a bike was pretty far gone. Like his gallbladder had already exploded. So and they were moving in his mouth. So I'm not really sure. Yeah, no one has really looked at that. It'd be interesting to like try to culture pentastomes, like put them on like a blood agar petri dish or something. See what happens. Yeah, but I don't think we know how long they live outside of the host. My guess is not long.
Yeah. And forgive me if we touched base on this earlier, but what are they feeding on within the lung? Is it just lung tissue? Is it something to do with, oxygen consumption or blood or what?
Yeah, they're feeding on the blood. So they are hematophagous.
Okay.
Yeah, so when you pull them out, or in that video where we were seeing them in the Neurodia, those little dark brown bands in them, that's snake blood that they're digesting.
Wow, that's crazy.
Yeah, that's why we think it likely causes pretty severe anemia, maybe more so in like a less active snake. But we're still figuring that out. One of my good friends and colleagues, Corinna Hazelrig, does a lot of blood work with pygmies that we catch at a mutual site. So it'll be interesting to see what she finds.
Yeah, that's fascinating.
She does a lot of hematology stuff with snakes and looking at how different pathogens, including pentastomes, impacts them. So it'll be interesting to see what she finds.
Very cool.
The SFD and pentastomes, for people that are herping and stuff, like how we went to Tallahassee and then came home and then went to Francis Marion and then came home, is there a known way as far as cleaning gear? Like what's the best stuff to do that with?
You're a carrier, Smitty.
So I think with pentastomes, I'm more concerned about people moving wild caught snakes in the trade, or accidentally moving cockroaches with like horticulture up the
Yeah.
coast, or moving small anoles like brown anole that made it to Boston a while back. Or so moving small animals I think is more of an issue. We actually don't have, like, there's not any studies about what percentage of ethanol or what percentage of isopropyl will kill pentastomes. I know for sure freezing does over time. Desiccation, like drying the eggs out, does not kill them. But the stuff that I if I'm moving in between sites, I just bleach everything, because I
Okay.
know the concentrations that and the time that it needs to be on there to kill snake fungal disease. So I'm more worried about spreading snake fungal disease than I am about pentastomes. Just because I think it would be hard for us to spread pentastomes. Like, just like we would have to like carry snake poop
Yeah.
with the eggs in it. And my guess is that there is adaptations for the poop to like perhaps taste better to invertebrates that eat poop when they have eggs in them. So it's probably
Interesting.
kind of like some type of attractant. This is just kind of speculation, but there's probably some type of attractant for invertebrates to eat the poop pretty fresh. But yeah, I think humans are more so moving it just by travel and trade of wild caught animals, and then just the animals that we're moving from South Florida up with plant trade and everything else.
Yeah, because we were talking about it in the group chat when I was at Harry's and stuff, and I don't know if it was Billy Jenkins or someone else who was like, I'm surprised that it's moved as as far and as much as it has, and I'm really not. I mean, you think about agriculture, especially like that alone, hitchhikers, be it pygmies or anolis or anything like that, I'm honestly surprised that it's not more common.
Hey, it goes back to our best friend, the forward fly.
That's right.
The fucking bane of our existence.
Yeah, and then just on top of all the imported animals that come in, and all the different shows and things, like it's really no surprise that they've gotten as far as they have,
Yeah, I mean, people sell wild caught Dorodia, and if like someone doesn't want it anymore, they're just gonna let it out outside. And they could have just brought that from Palm Beach County all the way up to Madison or something like that. And then we also we know Burmese pythons are like probably the number one culprit for the spillover event, but we're importing other animals all the time that carry them. So like Hemidactylus, like they carry them as larvae. Tegus have them, Tokes have them. So there could be multiple introduction fronts that we don't really know about or understand.
It's easy to point a finger at the big giant snake.
Yeah, it definitely is. And it's just confusing too, because it's like Burms don't really have high prevalence, and they also don't have high intensity. So they have pretty low infection intensity. And like I talked to Melissa Miller on the phone a few weeks back, and the prevalence is just not comparable to native snakes, which is fascinating. And there is a paper about tokay geckos in captivity. I forget which zoo from up north it was, but it was a zoo up north. They were wild caught, and then there was a handful of tokay geckos in Florida, and it was a good amount of them that had Relicella orientalis. And it's like another issue too is a lot of animals in captivity in the southeast US are held outside. And so they're exposed to whatever pentastomes are out in the wild, just because if there's, a tortoise hanging out outside in South Florida and there's cockroaches moving in and out of its cage, if that tortoise is infected, then it could spread to the environment and vice versa. So there's probably a lot of spread within, captive animals, both inside and outside.
Yeah, and I mean, Lisa brings up a good point too. As far as getting captives started on mice and not trying your best to not start stuff on anolis or any other reptiles or anything like that. But if you are freezing feeders and things like that, do you know how long they need to be frozen for for them to be effectively killed?
Yeah, that is a good question. I don't know.
I can't imagine it would be very long, because if the
Yeah.
blood's frozen and not moving, then they're not eating, and if they're not eating, then they're not.
Yeah, I'm guessing like 24 hours would be fine, even less. Like, I think once the animal is frozen,
Really?
it's fine. I don't know though. Like, I think we'd have to do some trials with that. The only trials that we've done with eggs, trying to like test their resilience, was desiccating them, so completely drying them out for I think up to like 16 days, and they were still
Wow.
infectious to cockroaches.
Wow.
Yeah, but we haven't really done something similar with freezing. I do know that like porocephalus eggs can be like in a refrigerator for a number of days and they can be fine. I don't know why they went for a refrigerator and not a freezer in that study. But yeah, yeah. There's just not a lot of information known, but I'm guessing that if you freeze them, you'd be fine. I mean, better safe than sorry. I would just wait a full day. Yeah.
Very interesting.
Hmm.
Yeah, it never even occurred to me about, the cleanup crew insects and invertebrates that are transmitters, and like I don't have isopods because I'm neurotic as hell. And even though I can't remember the specific species, but the quote unquote tropical isopod that we all have in our bioactive enclosures, they're found all over southern peninsular Florida. And everyone's like, well, I'm neurotic because I don't want to throw out dirty substrate and accidentally release an invasive isopod. So I have several friends who are like, well, look, the ones you find in your backyard, those are the invasive tropical ones. Just use those. And then God forbid you throw out, dirt that's dirty dirt that has these isopods. It's not that big a deal. And I still don't do it because I won't be that guy. Like I'm too neurotic. But that in itself, who would have thought that could eat infected poo and then go spreading it around?
Yeah, we don't have much information on isopods, but it could happen for sure.
Sure.
Yeah.
Sure.
Yeah.
Just like forward flies.
It's damn flies.
Those damn forward flies, man. Dude, I had one land in my cup at work like 2 days ago, and I just looked at it and I was like, you crypto carrying son of a bitch. And I just threw my drink away.
Not getting me. It's been documented in a handful of native species, though. I mean, garters, Nerodia, obviously, racers, I think coach whips.
Yeah, coach whips and black racers have the highest prevalence. But I can pull up the list here. Yeah, there's 18 total species of native snakes that have been found with adult infections, including one of the newest findings, which is a mud snake. We've got 3 different mud snakes from South Florida that we found with infections, which is fascinating.
Were they DOR or were they field collected and swabbed?
They were DOR.
Okay.
They were dissected. Mm-hmm. Yeah. So Colubric constrictor, Masticophis. Sorry, I'll use common names. So
you can go scientific.
You can do scientific. We're all about the nomenclature.
Okay.
Cool. Nomenclature is
it makes us sound at least somewhat smart.
Yeah. Pantherophis, Gutatus, and Alleganiensis, Drymarchon, Cooperi, Thamnophis, Sertalis, and Sordus. I'm just going species level here, not subspecies. Nerodia, we've got Clarkii with infections, Fasciata, Floridana, and Taxi, so the browns. And then Forancia abacura. We've got Lampropeltis, Elapsoides, and Gatula, and Floridana. We've got the coral snake, Mycurus fulvius, and we've got pygmy rattlesnakes, eastern diamondbacks, and Echistrodon with it.
So everybody.
Kind of everyone, yeah. 18 species is a good chunk.
I Have they documented as far as the eastern diamondbacks what those infections looked like in comparison to other natives?
We don't see a lot of infections in them. They more often have porocephalus crotali and just like a couple in their lungs. The first detection of it in an eastern diamondback, I think, was in Southwest Florida by Dr. Matt Metcalf. I think it was a paper in Herpetological Review. But yeah, I think in Southwest Florida is really the only place that we've seen eastern diamondbacks with Relicella orientalis, which makes sense because Southwest Florida is like South Florida overall, but I think because we have researchers in Southwest Florida that are dissecting a lot of snakes, we're just seeing that it's like very saturated there. Yeah, but we don't know much about the easterns, unfortunately.
Crazy. Very interesting. Scary.
Yeah, it is a little scary. It's definitely scary, especially when you find snakes dead in habitat, like we both have. Yeah, it gets scary. I think last year we found 8 dead in habitat, and this year I found 6, including yours.
And was it a similar situation where it was just a dead animal that just died?
So that's what I'm talking about when I say dead in habitat. So I guess 5 this year, because I won't count the one that was hit by a bicycle because that was not the Pentastomes' fault. But yeah, so I think one of the worst cases is kind of what got me a lot of followers on Instagram. There was a female recapture at one of my sites. I had captured her for the first time, I think, in like June or July, and she was doing fine. But then I caught her in the fall, and her snake fungal symptoms looked really bad, and she kind of just got worse. And then one day we were just kind of walking along. It was late December, and she was just belly up, which is obviously strange to see. And so, we tried to right her, and she wouldn't, and then we flip her back on her back, and she had very little response, very little writing response. And you could just kind of hear little puffs of air coming out. And so what we decided to do is just kind of sit with her for a little bit to just document what was happening. And then she just if you flip a snake over, you can usually see their little heartbeat. And so we kind of just hung out with her until that stopped. And then we just put her in a tube and started to sample her like we would otherwise. And then at that point, unfortunately, she was dead. I think that was kind of the most dramatic. Most of the time, every other snake I found has just been dead. Yeah. This one, it was definitely a little bit more emotional to see.
Sure.
Yeah.
Of course.
Yeah. Yeah.
Damn.
Yeah. So I think it's scary when it comes to that. And, seeing snakes, you see a lot of sick snakes. The more snakes you see, the more anomalies you see. And you just kind of, you're like, okay, yeah, I'm coming out here a lot, but what is the frequent, if I'm coming across, this dead animal just by chance, what is the frequency of how many are dying out here? And how many are due to this? Yeah. So it's definitely scary, but I guess motivating to continue the work.
Absolutely.
As far as the sort of the intermediate hosts, do they know, are frogs a heavier carrier over, lizards, or, roaches seem to be a pretty big one. Is there a main culprit in particular with any of those?
We haven't really done a lot of exploration outside of Central Florida. Dr. Terry Farrell and I are really the only ones that have looked at intermediate host infections, both in the lab and in the wild. But from what we've seen, southern toads can get hit pretty heavy, especially we were mostly like comparing, like for undergraduate research, like the difference between, Anolis sagrii and southern toads, and Axis terrestris, their infection levels, and then also kind of comparing, like, do southern toads at this retention pond have more pentastomes than this retention pond? And southern toads are pretty packed with pentastomes. But same with
the native ones. The
No.
native pentastomes? Oh, the non-native. Okay.
the invasive. Mhm.
Very interesting.
Yeah, from what we know, the native ones don't get into toads. But yeah, we've found wild caught intermediate hosts. We found brown anoles as carriers with larvae, green anoles, Cuban tree frogs, green tree frogs. I think we found it in a spade foot toad as well. Southern toads, of course. And then.
And this is all animals through dissection, or?
Yeah. Mm-hmm. Yes. And then which Plastiodon was it? I think it was the southeastern 5-line that we found some larvae in as well.
Very interesting.
Yeah, so it's mostly lizards and anurans, but I mean, I haven't dissected that many like mammals, shrews and stuff.
Right. The as far as like the genomic work that you're doing with the pygmies, what's sort of the goal with that?
I just spilled water on my computer. Hold on. It's fine. So I mean, my major goals, I feel like I kind of already said them, so I apologize for being repetitive, but I really want to understand how many distinct populations we have of pygmy rattlesnakes in Florida. From a previous study that focused on neutral genetics and more venom stuff as well, Dr. Mark Margus found that there's 3 distinct populations of pygmy rattlesnakes here in Florida, one kind of panhandle, one kind of peninsula, one in the Everglades. But I think that there's most likely more structure to the populations than what he had found just with his sampling. I'm pretty sure the grant that he had was more focused on like panhandle versus Everglades.
Right.
So there wasn't an incredible amount of sampling in the peninsula. And I think what I'm doing will kind of give us a better idea of what the structure looks like. And that's important to understand, kind of our genetic clusters of pygmy rattlesnakes here in Florida, and which ones might be more pressing to conserve habitat for. But I'm really interested in major histocompatibility alleles. So I'm interested in figuring out if certain alleles or diversity of alleles relate at all to the severity of diseases. So basically, like when we're talking about MHC or major histocompatibility complexes, it's just groups of genes and vertebrates that kind of code for different proteins found on surfaces of like cells and stuff of foreign substances. So there's MHC class 1 and class 2. And class 1 is more viruses, and class 2 is more extracellular stuff. So like snake fungal disease or Pentastomes. So I'm looking at major histocompatibility complex type 2 in pygmy rattlesnakes. And I'm also doing some stuff with water snakes as well, because there's some water snakes in Texas doing pretty bad with snake fungal disease. And we kind of, so we want to know if there are genetic underpinnings to susceptibility to these emerging pathogens. And no one's really looked at this in snakes in this capacity. There's one study on MHC class 1 in, I forget which snake it is, but it's just one study. And then there's a study in Cystoris catinatus of MHC class 2 as well. But yeah, so I'm really interested to see what we find. I've done a preliminary run, and it was just 17 pygmies from
Just 17.
from 6 different sites. Well, you need a good sample size to be able to catch the diversity, but we found 43 unique alleles just within
Wow.
those 17, which is really cool. I mean,
That's awesome.
we do expect like really high diversity because when we're talking about MHC alleles, they're under balancing selection, which means the more diversity, like you're selecting for more diversity within the population and within individuals. So it would make sense that we find kind of a high diversity of those alleles. But it might be that, if there's like with my advisor's research, she was able to find that there was like this one specific allele in frog populations that if they didn't have those, they would die from BD, from chytrid. So it could be something where there might be like a really strong allele that helps pygmies be able to survive this disease, or at least survive like the harshest parts of it. And that's something that I'm really interested in. So we might see kind of like a fixation of those alleles in populations that have kind of been not wiped out, but all of the more susceptible snakes have died. Yeah. But so that's kind of what I'm interested in when it comes to genomics of pygmies.
Are you going to incorporate all of the head patterning markings and photography into the genetic diversity survey, if you will?
Yeah, so that probably won't be like a chapter of my dissertation, but I'm definitely interested in seeing if the neutral genetic diversity relates at all to the different like patterns that we're seeing, because there's definitely differences between South Florida and Central Florida and North Florida and the Panhandle. Like Central Florida, we are so blessed. We've got big fat pygmies and they have incredible saturation in like the burnt sienna. So the stripes down the back, like, it's just beautiful, super high contrast. And then in like the Everglades, you are getting, or like in South Florida, you're getting like really light pygmies and like lots of speculation, just lots of speckling down the body and
polka dots.
Yeah, they're really cool. I really like South Florida pigs. I mean, you can get really light ones up here. They're just not as common. And then in North Florida, I find a lot of like darker pygmies with less
more purplish gray.
Yeah. On the West Coast, I find a lot of almost anery pygmies. So I have a site on the West Coast, and I find a lot of pygmies that just have like a dot of copper / like burnt sienna on their head, and then the rest of them is fine. I found one half striped pygmy, which was cool.
That's awesome.
Yeah, the latter third of its body had like not like perfect stripes, but they were like instead of having a dot or a heart or a chevron, it was just like squished all the way across on the latter third of the body, and it was
very cool.
It was very cool.
What vague locality are you willing to tell us?
The West Coast.
Okay. Fair enough.
Yeah, I mean, I'll tell you secretly when we're not live.
Good enough. I appreciate that.
Yeah, no worries. Yeah, and then the Panhandle just has, I don't know, those snakes are really cool, super small, but I do find a lot of like grayish blue ones, like almost, and like orangey too. It's like they almost look more like a Western pig, Streckeri.
Yeah, that's awesome, man.
Yeah, so I think it would be really cool to look at that. But yeah, I'm collecting, it seems like a massive amount of data, so it won't all be published within my PhD years, but I definitely think there'll be genetic correlations to the morphology. And I measure the snakes in a squeeze box. So they're below plexiglass in a clear bag, and we just kind of press them down gently. And so I have really good pictures of the entire snake's body and their design. So yeah, that's awesome. I try to get good lighting, but sometimes I'm out at night.
Yeah, still super cool.
And are you guys drawing blood for the DNA samples, or is it like a scale clipping?
Scale clip. Yeah. Yeah, I try to like limit the processing time so that one, the animal's less stressed, but two, I can find more snakes. So I kind of toyed with taking blood for a while, but then decided to do scale clips.
Yeah, that seems probably a little easier, especially if you're out and about, because
Yeah.
with blood and stuff, there's a whole storage component, I'm sure, and
Yeah.
Mobile lab.
yeah.
Yeah, I mean, you can store blood if you're just doing DNA. You can store blood in numerous ways. You can even just put it in ethanol.
I
But
have the Barbasol can with the little vials in it.
I have one of those.
You would.
I do.
Yeah.
Bought
I
it on
do.
the interweb.
Um, yeah, but just scale clips. Plenty of DNA in there.
Very
And when you're in these areas where you're finding high concentrations of pigs, is there a correlation to any other species? Like, how many corals are you seeing in comparison, like when you're in these areas with pygmies? Do you get a lot of other stuff as well, or is it?
Yeah, I've started to record the bycatch, and this is something that I wish I did last summer. But I mean, the study is going to continue for the next 4 years, so I'll get plenty of that data. But I do record bycatch now. I get a lot of cottonmouths, which is nice. A lot of racers, of course. Um, Ribbon snakes, garter snakes, bandeds. Those are kind of the most frequent. I have come across eastern diamondbacks, which is nice, but just road cruising.
Mhm.
And then corals are just kind of like once in a blue moon. I have a couple, like my more local sites, I see them more frequently at because I'm there, 2 days or, well, overall 6 days a month. And so I think I just have a higher chance of doing it. But
Yeah.
I've seen a couple at my far sites as well. It's just kind of random, usually first snake of the day type of thing. Yeah, it'll just
Mhm.
be first in the morning. Obviously on the move as always, but yeah, I do get quite a bit of bycatch, which is always fun. Yeah, and one of my favorite places to go is definitely South Florida. It's nice to get just a bunch of different stuff down there. I love getting lamps. Lamps are cool.
Have you found Brook's Eye? Man, that snake just eludes me like for years, 20 plus years. Still haven't found one.
I've only found one, and I just got it in March.
Okay, nice,
Yeah.
nice. Yeah,
Yeah.
there was a road down. I mean, we'll just ballpark it, call it east of Naples, right? Be as vague as possible. There's a road that went through a non-gated community. And when you road cruised at night in the summer after 9 p.m., you'd get almost guaranteed to find less than 10 snakes. And every single one was Brooks King. And every single time I've been to that road, I've never found anything. So I'm pretty sure it's a wild goose chase, but it's on the list. It's on the list.
I feel like the biggest thing that I've recognized about herping is just
Don't trust anyone.
well, that and like consistency and frequency. It's just kind of like the amount that you go out, what you put in, what you get out. And like sometimes I try to teach my undergrads. I'm like, guys, sometimes you just get skunked and it sucks. And you can put in all the effort you want in the world, and the weather is perfect, the ground temp's perfect, humidity is perfect, and they might just not be moving, or one just moved behind you. Like it's just like so kind of random at times. Like I do think snake hunting, especially when it comes to like understanding habitats, understanding behaviors, where you're going to find them and stuff, I think it does come with a little skill. And then also like if you're actually paying attention and like looking for the snakes, and when they're ambush predators, are going to be harder to see if they're not moving. But I think a lot of herping comes with a lot of luck.
It's 95% luck.
Yeah.
And it comes to time and field too. You know what I mean? You can't complain if you're only going once every other month. You gotta get out there and do it.
See, that's what Jake tells me, and that's what Katie tells me when it comes to my damn coral snake obsession.
Yeah,
and it's like people say that like you got to get out there, and it's like I have neighbors sending me pictures all the freaking time that live like
Yeah.
200 yards from me that are seeing corals on a regular basis.
Okay, so are you
And
taking that data and extrapolating the information that you need and then going for a walk twice a day, every day from
twice a day? I got a job.
Okay, so before you go to work, you take 20-30 minutes to walk a lap around the neighborhood. When you come home from work, just as the sun's setting, or even just as the sun's setting, go inside, have family dinner. Once everyone's wrapped up with dinner, go back out 8:30, 9 o'clock at night. Now you get the second round. Right as the dews start to come
So
out.
what I have gathered from all these sightings that were not mine is that the corals,
That God
at
hates
least
you.
here in my neighborhood, people are seeing them mostly between 10 a.m. and noon.
Really?
Yes.
See, I would never have
Which
thought
I
of that.
also
That's
thought was
crazy.
strange.
That's so wild, man. It shows you how much we don't know.
They are considerably more diurnal than I sort of initially thought. But
I
they're
mostly see them.
everywhere.
Yeah, I mostly see them earlier in the morning, like between 7 and 10. But it's hotter here, so
Yeah, I don't know. It's like it's to the point now where me and Jake were cruising over the weekend, and I was like, dude, I'm actually to the point where I'm getting like legitimately pissed because I have people that live down the street from me posting in the South Carolina Snake Identification Group.
Yeah.
I've seen 6 corals this year. And I'm like, are you freaking kidding me? Like, the funny part is, Phil, I don't know if I told you this, but there's the one guy that keeps posting in the ID group. And this is a statewide group, right? And he keeps seeing corals. He found a really nice copperhead the other night. Like, that's cool, but I'm whatever. And so finally, I noticed in the comments he said he was on Ladies Island. So I finally messaged him. And I was like, hey, man, like, you ever need anything removed or just relocated, whatever, like, let me know. And I was like, out of curiosity, where on the island are you? That dude is 30 seconds down the street from me. Not even kidding. He said the street, and I was like, that sounds really familiar. And I was like, looked it up on Google Maps, and I was like, are you fucking shitting me? Like, I could walk to the end of my street through the woods, and I'm on his street. Like, it's right there.
Well, just remember, anytime you are getting frustrated or annoyed or rageful about the fact that the coolest North American elapid eludes you perpetually, remember that I'm in the same boat, bro. The only ones I've ever seen outside of captivity were ones that people caught and I released. So
yeah, but then I also
have you not seen a Sonoran?
I have seen a Sonoran.
Yes. Yeah.
Those ones I feel like are easier.
Well, I've only found one, and that's because I've only been to Arizona a handful of times. So
yeah.
I mean, it's just super frustrating because like there was last year, I think it was, or the year before last, I take a walk literally down my street around the corner, like 30 yards, and there's 2 corals within like 15 feet of each other that are road pizza.
Oh,
and I'm like, are you like, I got to the point where I thought maybe someone was messing with me and was like just putting dead corals on the street and like,
I won't lie, I was
like, I'm getting to this weird conspiratorial like tinfoil hat levels of this is too coincidental to keep happening. This like someone's got to be messing with me, and it maybe even be Jake. Who knows?
I was legit at Petco getting fish food, and I found they have all these like new rubber toy animals for children, like educational rubber toys. They had a coral snake. I was going to buy it and like stuff it in a manila envelope and just mail it to you just to be a dick. It was like honestly, it's the perfect one. And then I thought, I was like, nah, that's kind of dumb. He's gonna laugh at it and go, whatever. And then I was like, I'll keep it in my pocket. And then next time we're hanging out, just like throw it in the grass and see what you do. Yeah.
I like posted, I asked Jack Vicente, I was like, what did I do? Like, why are you doing this to me? Like, what is use your
did
juju?
You actually ask Jack Vicente, like the Jack Vicente, if you're fucking coral cursed?
Yes. I was like, what am I doing wrong? It's like, Jack, tell me. Like,
I can't believe you. What did he say? What did he say?
He was just kind of like, it is what it is, man. And I was like, that's not the answer I want.
That is the answer, man.
It is the answer.
Yeah.
You know, that's what like the whole thing boils down to is everyone's like, you got to get out there and you got to look. And it's like, Gary down the street doesn't have to look. My neighbors around the corner don't have to look. Like,
yeah, this reminds me, like, So me and my colleague Xander really want to see male-to-male combat and pygmies. We go out all the time. I just told you I'm out 5 days a week. Especially during the breeding season, we go crazy. We have not seen that at all. No
Is
matter
it even
how,
a thing?
yeah.
Is it really a thing?
Male-to-male combat and Cistoris, yeah.
Midget wrestling, basically. Dude.
All right. Well,
It's
you
basically
know what?
a little like thumb wrestling, but
Okay,
there's
so
names
when is this male and male combat in Cistrus supposed to happen?
in the fall. So,
All right,
well,
so-
Cistoris malaria. So it's going to be like they start breeding like September, kind of like.
All right, so in like August, September, I should put two boys together and record it for you and see what happens.
Yes,
All right, because
you should.
I never had a reason to put two boys together, but you know what? We're doing it in September.
They-
That's what's
Start
happening.
placing bets. Can you like live stream it and we'll like
Yeah,
We'll
that's
see.
fine.
Yeah.
on
So
DraftKings or something?
these people come up to me at my site on Friday and they're asking me what I'm doing. And it's just kind of like older people. And they're like, "Oh, you study rattlesnakes." Well, I forget what state they were in, but they were like, "We saw timber rattlesnakes and they were dancing on the side of the mountain." I was like, "Are you kidding me?" I was like, "When was
Jesus.
this?" And she was like, "Yeah, someone told us to like come watch and it happened for like an hour." And I was like, and they were like showing me what they were doing. And I was like, "You saw male to male combat." I was like, you're so lucky like that happened. And they were like, "Wow, look at us go." I'm like, "What do you mean?" I hate
Yeah,
you.
I didn't
Wasted
think-
on you people, man.
I didn't think Cistrus did it. That's wild.
Yeah, so Xander Perlman, he's at the University of Illinois and he's studying Cistoris catenatus and Cistoris malarius, and he does a lot of stuff with like foraging behavior, but he's also looking at reproductive behavior, and he's doing some studies with captive catenatus at the Toronto Zoo, I believe it is. And so he initiates like the males having combat and then films them and stuff. It's really cool. Yeah, he's got a lot of really good video
That's
of
awesome.
it. Yeah. Yeah, they do it. They go up and they like push each other down, the whole
The
thing.
herb nerd equivalent of bum fights.
Yeah, legit, man. That's awesome. But no, all joking aside, I'm gonna put two boys together this year and see.
I can ask Xander for more specifics of what he does
Yeah.
to like get the catenatus to start fighting. But
Duking it out. And I wonder if honestly, I wonder if I got a winner if they would breed that much better.
they might, right? It stimulates.
It wouldn't
Yeah.
surprise me at all.
Yeah. Yeah.
Yeah,
I mean-
and the same thing locally goes for the Eastern Diamondbacks too, because there's a state park here that apparently has a nice population and a like perfect habitat. And someone posted the other day on one of the trails an Eastern Diamondback, and they're like, "I was terrified," and I'm like, "Go to hell,"
Nice.
because I know people that have been going out there for like over a decade. And they're like, "I've never seen one out there.
Wild.
It's crap," and I'm angry.
You have the right to be angry. My field techs know that if we don't find a pygmy within like an hour, they're like, "Oh no, Jenna's gonna get
It's
grumpy."
turning. Yeah.
I start to there. I'm like, but now like the ones that come out with me so much, like as soon as we don't see a pygmy for like 30 minutes, they start getting pissed, and I'm like,
30 minutes. 30 minutes, Smitty. You hear that? 30 minutes. Do you
Okay,
know
well,
how many thousands of miles I put on my vehicle in
okay,
search of pygmies?
same. I put in an incredible amount of effort.
No, I know, but you're going 5 days a week. I'm going like twice a month. So
In South Florida, in one weekend, we walked like 36 miles and we found one pygmy.
Damn.
where in South Florida? Ballpark.
Everglades.
Like Everglades National or like STAs?
Everglades National.
Yeah, see, that's tough, man. I've never found a pygmy down there because it's just too wet.
It is tough. There are really only very like select locations that you can find them in the park.
Yeah, and I've only ever found in that central core where it's Everglades National that goes straight up north and hits the STAs and goes up to Lake Okeechobee into the agricultural area. I've only ever found one pygmy in that column, and it was right where the Indian reservation meets the STA. And I'm convinced that it was essentially coming off the res where it's a much higher elevation. I mean, I say much higher, it's 8 inches higher, whatever it is. And and that thing was gargantuan, one of the biggest pygmies I've ever seen.
Yeah.
So,
The biggest pygmy I ever got was from the Everglades. It was 175 grams. That thing was a
I know.
beast. He was almost 3 feet. Like he was
Yeah.
huge.
yeah, that's
Massive.
this one was probably close to that.
Yeah.
And again, I just feel like I don't go to Everglades National because it's a bit of a hike for me, and there's just so many humans. It's so many humans,
It's pretty
and
gross.
I feel like and I feel like it's just too wet. So I mean, I have friends like our friends Jake and Skylar and Preston. They're going to be going down there. I'll probably link up with them, for a night or two and see what's what. But pygmies, man, you got to go to Sandland. You got to go up by you.
I don't know. I've been finding pygmies down there. Maybe you're just not good
in Everglades National. Yeah, see,
Yeah.
I just don't go enough.
I mean, it takes an incredible amount of effort.
Yeah.
Every time I go, it's an incredible amount of effort, and yeah, it's not easy. Like I remember we caught one snake and I had like the biting fly bites on my eyes and they were starting to like swell.
yeah.
And I'm like holding this little pygmy. I'm like, I can't see. I'm like, this is horrifying. And we had one person with us that was smoking cigarettes, and that was like literally our saving grace because mosquitoes don't like that while we were processing. And like there was times where like it was so bad that we just processed the pygmies in my truck.
Yeah.
Like this is horrible. Yeah.
Gotta get Thermacell.
The Thermacells do help. I do have one. Those really do help. But the Everglades is
rough.
Is a different beast for sure.
A lawless swamp.
Yeah, I do love it though. It's just a whole different world down there. You don't have service. It's just kind of nice. But yeah, no, the Everglades is crazy. And I remember that it was like 2 weeks after I left the Everglades, someone posted on Instagram that they saw 2 males in combat where I was searching. And I was like, oh, you just gallivanting through the Everglades and you get to see that? I'm like,
yeah,
I walked
horrible
30 miles and didn't see that.
I'm with you. I'm with you.
Yeah. So although we get frustrated within 30 minutes, we definitely stick it out. Like last weekend, we cruised for more than I'd like to say.
Yeah.
For 4 pygmies. And yeah, I had a researcher reach out to me and was like, hey, like I went out to the same spot and find any pygmies. I was like, how long did you cruise? Because my ass was cruising like 7 hours.
Yeah.
Yeah, just constant. Yeah. But
when you do that, I mean, when you are cruising them, are you finding them like later into the evening? What seems to be is there a pattern there at all?
You want all my tips right now?
I just want to know what I'm doing wrong.
Yeah, I feel like pygmies really, I was actually looking at this data the other day. Pygmies really like to move on that. I think this is like snakes in general, but pygmies for sure like to move like an hour and a half before sunset up to sunset, and then like sometimes just after.
Yeah.
In South Florida, lawless, like you're saying. Yeah. I think you can find them probably into the early morning, and you just have to go crazy in the Everglades. I pretty much sample nonstop when I'm down there. But everywhere else when I cruise, it's usually like right before sunset.
Oh, this is the video that Lisa sent?
Yeah.
Excellent.
Would go full screen.
Yeah, I would say in my area, hitting the agricultural areas and stuff, it's like you said, about a half an hour before the ball of the sun is actually below the horizon line to say maybe an hour after, if that.
Yeah, exactly. I do. So in the past 2 weeks, I have found pygmies interacting with each other. And this is the first that I've seen besides finding a mom with her litter.
Mm-hmm.
And so 2 weeks ago, I found 2 pregnant females on the move together.
Oh, wow.
And they were like headed towards the same sable palm. It was very interesting. I was like, what are you guys doing? I just think there's a lot more sociality when it comes to vipers than we like to think or understand.
Absolutely.
And then this past Sunday, I found 2 males just hanging out, like both stretched out just next to each other. And I'm like, what?
You think that's like the ideal cryptic bass gang, or?
Do I think what? Sorry, I was watching.
Do you think it's like the ideal basking spot? Maybe they both were attempting to cryptic bask, and it just happened to both be the perfect amount of sunlight. You know what I mean? Or do you think they actually were interacting?
It's the males, I think, is harder to say because one was like in a loose coil in front of a log, and then the other one was stretched out behind the log. So
Okay.
that was probably like that. The male that was stretched out was probably not even recognizing that the other male was there. So I would say that was less of an interaction, but the two females were like on the move together. So that was really cool. I've definitely seen like associated pairs as well, which is really neat. So like a male and a female hanging out or like coiled next to
Have
each
you
other.
seen copulation?
I have not seen copulation myself, unfortunately. Not yet.
A friend of mine from work, he lives in, oh man, I think it's Martin County, so Port St. Lucie area, and his house, his backyard butts up to the WMA, and he's a big hunter, and he literally walks out to the WMA every morning to go hunting, and he was posted up on a tree just kind of hanging out, seeing what the morning holds, and a pygmy starts to slither up to his boot, and he kind of just stands still, and he's not afraid of snakes. He enjoys it. He likes them, and he knows I like them. And he took out his phone. He started recording it, and as he's recording it, he realized that one snake that was slithering towards his boot was tethered to another snake. So the female was just dragging the male, and they were totally locked up. It's crazy. Once in a lifetime.
Crazy.
Yeah.
Yeah, I know at one of my sites, the park rangers have like sent me pictures of like them locked up like in the middle of the trail. I'm like, why can't that happen when I'm there?
Yep.
Yeah.
It's our curse.
My excuse is I just tell Jake, I'm like, God hates me. That's all. I did something at some point and I'm paying for it. Don't know what it was, but picking up the tab.
Yep. We got to work harder, Smitty. That's just it. We got to grind more.
Gary doesn't have to grind. Old neighbors don't have to grind.
Yeah.
I gotta grind. He posted one. I'll send you the picture, but it was like the yellow on this thing was just insane. Like it had this weird, almost like a spray painted look to it, and the yellow bands, and it was dead. I don't know who killed it, but he said it was a neighbor, so it was someone I live near. It was a really nice looking coral, and I'm like, son of a bitch.
Yep.
Yeah, the one I found last week had like 2 circles on it, which was really cool. I had never seen that before. Yeah, I think it was a female, and she just had these 2 really cool circles on her.
Well, we're at the 2-hour mark. Phil, did you have anything else?
No, we covered pretty much everything that I wanted to bring up. You know,
plenty of stupid questions at you.
Yeah, plenty of them.
They weren't stupid.
Got to figure out how to weasel my way into a survey next time you're down south.
You don't have to weasel your way. You're welcome. I mean, I don't really trust your eyeballs from these conversations. But
That's good.
Maybe you'll walk in the place where they aren't, so I can walk in the place that they are.
You know what it is? I'll just follow you, chain smoking, keep the mosquitoes away.
Perfect. Amazing. Yeah. I probably will be in South Florida at the end of this month.
Oh, cool. Excellent.
Yeah. If anybody sees anything or notices anything, you want information.
Yeah, correct? If you see snakes with worms crawling out of their mouths, you can hit me up on Instagram, but you can also go to Slam's website or Slam's Instagram. Slam's website will lead you to there's a contact page, but there's also a reporting page too. So you can directly report sightings of what you might think is a lungworm on EdMaps. And so that link is right in our website. And yeah, that'll be super helpful. Of course, we have to take that data with a grain of salt. But if we do see okay, wow, this person posted in Georgia that they saw this racer with a lungworm crawling out of its mouth, it appears to be the invasive one, then we'll kind of hit that area a little bit heavier and get more information and investigate over there. So we won't kind of like sound the alarm because we need to actually see that parasite in person. But if you are a herper, you can collect dead snakes. I can send you, if you're not on like federal land, of course. But you can collect dead snakes, and I'll take them and dissect them. And if they're in good shape, they'll go to a museum. So that's pretty cool. But yeah, and if you're doing permitted research where you're already handling snakes, we can talk about how to detect them in fecal samples and cloacal swabs.
Excellent.
Yeah.
Excellent. Do you want to give the Instagram that they can hit you up at?
Oh yeah. So it's my main Instagram is fl.samiba. So it's kind of like my species code for Cistoris malarius barbari. So fl.samiba. And then Slam's Instagram is slam.conservation, I think. We're still working on kind of getting that more live, but yeah.
Cool.
Yeah.
Excellent.
Yeah, we really appreciate it.
Yeah, it's a great show.
Yeah, thanks for having me on. Let me know if anyone has any questions. Happy to answer any.
Awesome.
Yeah, I'm sure at some point we'll do like an update or something in the next, year or whatever. And if there's any new developments, let us know, and we're happy to spread any information you guys need us to or anything like that. Just let us know. So this episode was brought to you by BlackBoxCages.com. Check them out. Facebook, Instagram. Use that code THN to check out. Get 10% off your order. Then hop on over to FulviusApparel.com. I have to name it after Fulvius because I'm never going to actually see one. So this is how I get my fix. Use the code THN at checkout there as well. Get 15% off your order. But right now, buy one, get one half off on everything on the website. You can't use it with the THN code, but that is going on until the 11th. So take advantage of that while you can. Cold-blooded caffeine, grab some coffee. That Snakes and Stogies blend is where it's at. But any of the blends they offer, you will not be disappointed with. And Trey and McKendrick are just cool dudes. So support them.
Pinky's out.
I have no idea what's going on Thursday. Jake's going to be out of town, so there's not going to be any flipping tin. Maybe I'll see if Chris and Harry want to do a king snake thing again. Otherwise, we're just not going to have a show Thursday, but we'll see.
Fair enough.
Been taking a lot of breaks on Thursdays lately. I feel like we've been slacking.
Well, you got to get Jake and Sky to get their shit together.
Yeah, Jake's going on vacation or something. What a nerd.
What a fucking nerd.
What a loser.
Is he herping? No.
He's probably going to get some in there. He's going to Tennessee somewhere. Like Pigeon Forge, I think is what he said. So
nice.
I guarantee you he's going to poke around somewhere. At least
Salamandra.
Yeah. So thank you, everybody. We will see you later.
Bye.